Welcome to Falcon Physician Reviews. This is the online course for genetics lectures for the Step 1 USMLE. My name is Nathan York, and I have a PhD from the University of Texas Southwestern Medical Center at Dallas, and I will be conducting this lecture series. Let's begin talking about definitions and tools of genetics. Let's begin talking about a few definitions and tools of genetics. So genetics is the study of hereditary traits, or in other words, human characteristics that can be passed from parent to child. An example of this could be a child's eye color. It's a physical characteristic that is determined by the eye color of the parents. In medical genetics, we're particularly interested in clinically significant traits, something that causes a disease or something that may make somebody more prone to developing a disease. A gene is the basic unit of heredity, more specifically a segment of DNA that determines a particular trait or causes a particular disease. Most genes are transcribed into a single mRNA, which is translated into a single protein. As you can see from the diagram here, we have the RNA polymerase reading the DNA and constructing a strand of RNA. That RNA is then read by a ribosome to make a protein. As far as the USMLE Step 1 is concerned, you should always assume that you have one gene for one mRNA for one protein, unless it is otherwise specified. The genome is the total DNA content of each cell in an organism. Normal human cells, with the exception of gametes, contain 2 copies of the entire genome. This means that there are 2 copies of each gene in the human genome. There are approximately 3.9 times 10 to the 9th base pairs in the human genome, which make up approximately 27,000 genes. Now, a chromosome is a piece of DNA coming from the genome. So the genome is divided into discrete long chains of DNA, and we call these chromosomes. Humans have 23 pairs of chromosomes for a total of 46 in each cell. 22 of these chromosomes are called autosomes, and then there's also a pair of sex chromosomes consisting of either 2 X's or an X and a Y. Now, during cell division, chromosomes are condensed into the structure that you see here, and we have some more terms that we can memorize. The centromere is the piece that holds the 2 sister chromatids together during mitosis or meiosis. Telomeres are the ends of the chromosomes, and then we define them by P arms and Q arms, or short arms and long arms, depending on the position of the centromere. A locus is the physical location of a particular gene, or in other words, the address of that gene on its given chromosome. When we take a chromosome and we can stain it with certain stains, we create banding patterns, and then we number these banding patterns, which we then use to define a given locus for a gene. Each gene exists at a numbered locus, as you can see here. For example, 1p33 or 1q23. So that would be a gene located on chromosome 1 on the P arm at position 33, or again chromosome 1 on the Q arm at position 23. An allele is an alternative form of a gene, usually caused by a difference of one or a few nucleotides. For example, aldehyde dehydrogenase is an enzyme involved in ethanol metabolism. The major allele, ALDH2-1, has a glutamate at position 487. There's another allele, ALDH2-2, that has a lysine at that same position. It has a lower activity. This mutation is responsible for alcohol intolerance in about half of individuals of Asian descent. So again, here we see it's the exact same gene. The gene is aldehyde dehydrogenase, but there are different versions of that gene, and that's what we refer to by alleles. A genotype is a description of the alleles that an individual carries at a particular locus. Using our aldehyde dehydrogenase gene again for an example, if the individual carries the same allele at a given locus on both chromosomes, then we call them homozygous. So here we see chromosome 12, the Q arm, which is where the location of aldehyde dehydrogenase is, and this particular individual has ALDH2-1 on both sections or both chromosomes. Therefore, they would be homozygous for this gene. Now, if the individual carries 2 different alleles, they are termed heterozygous. In the example here, we see he has one chromosome that has ALDH2-1 and another chromosome that has ALDH2-2. So he inherited different alleles from his parents, and he would be considered heterozygous. Now there's another definition that you need to know, and that's hemizygous. We most typically talk about this when we talk about males. And remember in males that they have two different sex chromosomes, one X and one Y. There's only a single allele at each locus, right? Because X and Y are different chromosomes, they'll have different genes on them. And so at each locus they will only have one gene. They won't have a companion gene on a different chromosome. So they're considered to be hemizygous for each of those genes. For example, here in the diagram, we see the SRY gene on the Y chromosome. There's no counterpart to that gene on the X chromosome, so the male would be said to be hemizygous for that gene. A phenotype is the visual or functional consequence of a person's genotype. For example, a person with brown-brown genotype at the BEY gene locus will have brown eyes. That's their phenotype. So the brown and brown would be the two alleles that they have. The BEY is the gene, and their phenotype is brown eyes. Now a person that has blue-blue genotypes at the same gene will have blue eyes. Now let's discuss a few of these genotype-phenotype relationships. There's different things that we can see happening here. For example, with a dominant allele, what you have is an allele that expresses its phenotype in either the homozygous or heterozygous state. In other words, as long as you have one copy of that allele, that will be the phenotype that is expressed. A recessive allele is an allele that expresses its phenotype only in the homozygous state. For a recessive allele to be expressed, you have to have two copies of that allele, or be homozygous for that allele. The example going back to the eye color, BEY gene, brown is dominant, blue is recessive. So if we look at the three examples here, if somebody has two dominant alleles, brown-brown, they will have a brown eye genotype, or brown eye phenotype. If they're heterozygous, brown and blue, then again they express brown. Their phenotype is brown eyes. So that would be, again, the dominant allele being expressed over the recessive allele. And look at the third example, blue-blue, blue being the recessive allele, somebody that has to be homozygous in order to express that phenotype. A couple of other definitions to know, codominant alleles. These are alleles which both express their phenotypes in the heterozygous state, so there's not one that dominates over the other. A good example of this is in considering the ABO blood group and AB antigens on the surface of red blood cells. A and B are genes for antigens that are expressed on the surface, and they're both dominant. O is a recessive phenotype. Therefore, if somebody is AA or AO in their genotype, they will have an A phenotype. Or if somebody is BB or BO in their genotype, they will have a B phenotype, meaning they will express that antigen on the surface of their red blood cells. If they have an OO genotype, that would be an O phenotype. Again, we see the homozygous recessive phenotype being expressed. Now, if somebody has an AB genotype, or they're heterozygous for A and B, they will express both A and B equally. And these are the people that have AB type blood. They're both expressed, one doesn't dominate over the other, and we call this codominance. Okay, one of the tools that we use in genetic analysis is a pedigree. A pedigree is nothing more than a family tree that's drawn to ascertain inheritance of particular traits. And here we have an example of a classic pedigree. Now, for the most part, there are certain things that are very universal in pedigrees. Squares represent males, circles represent females. Most of the time, a shaded individual would be an affected individual, and if it's half shaded or perhaps with a dot in it, that expresses some kind of carrier state or something like that. However, it's always important in going through your USMLE step 1, all of the pedigree questions that you have, to make sure you look at the legend so that you don't mistakenly misidentify something. A couple of other things just to point out here. When you see two horizontal lines between two individuals, as you see in generation 3, individuals 6 and 7, that indicates consanguinity or an incestuous mating. A lot of times that's a good clue for a autosomal recessive phenotype. What you can also see is if you have an angle coming out like you see in generation 3 with individuals 1 and 2, that indicates twins. If there's a horizontal line connecting the two as you see in generation 4, 1 and 2, that indicates monozygosity or identical twins. Another tool that we use that you should be very familiar with is the Punnett square. We use this to indicate the probability of a particular genotype in the offspring of parents with a known genotype. So here we see on the outside of the square two heterozygous individuals, and then by combining each allele, we can therefore assign probabilities that they will be homozygous dominant, heterozygous, homozygous recessive. You should become very familiar with this. Now, we see for two heterozygous parents there are 4 possible offspring genotypes depending on which allele the parents contribute to the zygote. Again, each parent has one dominant A, one recessive A, and so depending on how those match up will determine the phenotype and the genotype of the offspring. Each square represents a 1 in 4, 25% chance or probability. Therefore, 1 out of 4 offsprings will be homozygous dominant for the A, 1 out of 2, or 1/2, will be heterozygous, and 1 out of 4 will be homozygous recessive. For the USMLE Step 1, this type of Punnett square, this single cross Punnett square, is going to be the most useful to you. If you think you see a question that might require a double cross, don't try to map that out. It will take too long and you don't have time to answer that type of question. So think about another way in which you can answer it. And we're going to give you a few hints a little later on that will help get to those types of questions. But single cross Punnett squares are definitely your friend. That concludes this section of the lecture. In the next section, we'll cover basic Mendelian inheritance and single gene disorders.